Good morning. My name is Jeannie Montblanc, and on behalf of the Great Basin Fire Science Exchange team and our partners, I would like to welcome you to this webinar titled Using Field Studies to Find the Most Promising Seed Sources for Restoration, presented by Beth Leger, plant ecologist at the University of Nevada. Before I introduce our speaker, I want to let you know that you may ask questions of the speaker or me at any time during the webinar by typing your questions into the questions pane of your control panel located at the top right of your screen. I will keep questions for Beth in the queue and field them after the presentation. I also want to let you know that whatever you do in your control panel does not affect the webinar presentation, so you're welcome to type a test message or test your audio at any time during the webinar. If you're having problems with your audio, please open your audio pane and check your audio selections. Now I would like to introduce our presenter. Beth Leger is an associate professor of plant ecology at the University of Nevada in Reno. She studies local adaptation and rapid evolution of native and invasive species and is interested in how genetic variation affects the distribution and abundance of species. She works on projects that gather basic ecological data about rare species, natural communities, and invasive species control, providing information that can assist management and restoration decisions. She co-directs the Museum of Natural History at UNR, where students and faculty do K-12 outreach on native biodiversity in the Great Basin. Welcome, Beth, and thank you for being here today.
Great, thanks, Jamie.
All right, I'm making you the presenter.
Okay. I think that's good.
Yep.
See my slides?
Yep, looks great.
Okay, great. Well, thanks, Jamie, and thanks for all of you for joining me. I'm going to be talking today about just what my title says. I'm going to be talking about how we can use field methods from ecology and evolution to inform seed source selection for restoration. This is kind of going to be a summary of things we've been working on in my lab for the last decade. I'm going to be talking about a lot of work of postdocs, graduate students, and undergraduates in my lab focused on solving these problems. I've given various versions of this talk, shorter versions that I often do a lot of telling and not showing. So this time I'm really going to bring a lot of the data to the table and show you graphs, hopefully not in a painful way, to demonstrate just what it is I'm talking about. So the outline, let's see if I can switch slides. I'm going to talk first a little bit about restoration in the Great Basin and some things that I learned when I came here from California. Things are quite different from where I came from, and that was really eye-opening for me. And I'll just talk about that a little bit. The bulk of my talk is going to be about what I've learned from these field experiments. I'm going to talk to you about some methods of measuring natural selection during restoration, which can be really informative. I've also done work looking for evidence of evolution in response to invasion, and also quantifying traits that help plants establish, especially in the driest, most invaded parts of the Great Basin. In the end, I'm going to do a little short shift on this because I think I want to put more science in. Maybe questions can address implications and future directions more than I'm going to do here. Okay, I'm not going to spend much time talking about the cheatgrass fire problem in the Great Basin. I'm going to assume some familiarity with that with this audience. But we know that in these systems, fires can be devastating, especially when we lack a perennial understory to respond. We can transition from shrub and perennial dominated systems to annual dominated ones, sometimes in the course of one fire. And it's this level of cheatgrass density that you see on the right poses serious barriers to establishing perennials from seed. That's not to say that we don't try. After these really large fires, that's actually not a bad time for action. You can lose a lot of the cheatgrass seed bank depending on the timing and temperature of the fire. This post-fire, kind of looks like the surface of the moon, but this can also be a window for action. There are lots of efforts by federal and state agencies to seed these areas after fires in an attempt to prevent them from transitioning from perennial to annual systems. So one of the things that I was blown away by when I moved here from California was just the sheer scale of what's going on in the Great Basin. So this is data from the BLM Public Land Statistics from Nevada alone between 2002 and 2012. And you can see the acres treated and dollars spent on emergency fire stabilization and rehabilitation projects. As a Californian, I have to say this really was sort of shocking. I was used to restorations much smaller size, and you can see here these are astronomical numbers. You can also see there's a cyclic nature to these fires. 2006 is a really wet year, then 2007 and 2008 we have a lot of birds. 1999 was similarly a huge year, but you have these periods of drought and then wet and then fire and then we're still in drought. But anyways, these numbers are phenomenal to me. So this is the desert, and not all of these seeds grow. Failures are all too common. There have been other webinars that have addressed success of some of these rehabilitation projects. This is a south aspect in a very dry area. And you can see here there are all these plants growing in rows. Well, those are not the seeded perennials. Those are cheatgrass very happily taking advantage of these drill rows. And this is all too common. You can end up with rows and rows of cheatgrass and very few seeded natives. So why is that? I show this to my students here, and I just have to remind them that, you know, this is the desert, and the first reason why our success rates are low is that not every year is a good year for establishing from seed in the desert. It's just not the way these systems work. If it doesn't rain, that's not going to be a recruitment year, and there's absolutely nothing we can do about that. So, you know, what level of success should we expect? Some people are talking about building that into our restoration treatments. We can't just assume that every time you put a seed in the ground, it should turn into a plant. Sometimes it's too dry. So we can't do anything about this. We just need to recognize that that's true. But what we can maybe do something about is the second point. We can think about the seeds that we're putting in the ground in these huge seeding efforts and think about if we're using the best seeds for our situation. I suspect, or 10 years ago suspected, now I kind of know, that there was a mismatch between the genotypes that we were using and the environments where they were being planted. And I'm going to spend a little bit the next, I don't know, 5 or 10 slides showing you why I think that. Okay, so first of all, again, very Nevada focused. We are the center of the Great Basin, so I feel pretty okay about being Nevada focused. But I'm going to tell you some information about where Nevada's seeds come from. The next slide is from the Land Treatment Digital Library statistics, looking at available data on seeding treatments in Nevada between 2006 and 2009. This is a really grass-focused process. Mostly native grasses during this time, less native shrubs and other types of species. But this is a super grass-focused effort. There are reasons for that. Grasses establish really well from seed, unlike some of these other functional groups, and so they are a logical way to reclaim some of these areas. There is, of course, more interest in using native forbs and shrubs in these situations, but they are trickier to establish from seed. So grass has been really the focus. So this slide is showing, this is one of my favorite slides. This is about the 8 sort of workhorse grasses that are often seeded in the Great Basin. And you can see that these red dots represent sort of named cultivars or named releases that are part of that slide I showed you. The seeds that we purchase are often, almost entirely, one of these or many of these cultivars. I'm not explaining that very well, am I? But basically, the point that I like to make from this slide, if you are a Nevada resident, is that there's only one cultivar that's sourced from Nevada among all of the plants that the Nevada BLM and other agencies have to buy from. If you live up here in Idaho or Oregon or Washington, this might work for you, but I have to tell you, down here in Nevada, we feel a little bit lonely, and we could use some more of these. You know, Basin wild rye grows all over Nevada. We really don't have to be buying seeds from Canada, or we would prefer not to. If you know anything about local adaptation, you know that a plant up here in Canada who's experiencing 30, 40 inches of precip a year is not expected to do well when you put it down in these 4 to 10 inch precip zones of the Great Basin. This is more than just an academic complaint. I'm going to show you some data from a site where we did some restoration in Mason, Nevada, very, very dry, and the plants the native grasses that we bought are listed down below. Just like everyone, this is when I first got here. I didn't have access to local plants, and so we bought these guys and put them in the ground. We knew that in this super dry zone, you can't just put plants in the ground and expect them to establish. We had access to water. This is in the Walker Basin, and we had access to irrigation. And so we thought, well, maybe we can give these plants a really good establishment year let them get established and then turn the water off. And maybe even these non-local genotypes will be able to persist. So what you can see here is at the end of 2 growing seasons where we irrigated these plants. This is May. Keep an eye on this tree. I'm going to show you some pictures with this tree in the background. I think you can say that we were actually probably too successful. These densities are really, really high. But we have a good stand. We thought self-thinning would occur, and we could maybe keep some of these grasses on the landscape. This is the next year after a growing season where we turned the water off. You can see there still are some native grasses in here. They're pretty dry, but we hoped that they were just senescent and not dead. But when you go out there, again, here's our tree. When you go out there the next year, those plants, it's as if we never planted anything. They're all pretty much gone. And so what we learned from this is that even if you give these non-local genotypes the best establishment conditions in the world, they still can't hack it when you turn the water off. We are in the process of repeating this experiment with local grasses. There are grasses out here and shrubs, so we've done some collections, and we're trying to do less water and local genotypes and see if we can get some establishment in these sites. So as mentioned, these aren't just academic concerns. These are true issues about the suitability of some of these cultivars to grow in dry places. Another point about these dots, it's not just that they're from wetter places than where we in Nevada are trying to plant them. It's thinking about the reasons why a particular population was selected for use. When I started, again, moved to California, tried to wrap my head around this issue, wondering what is it about this particular spot that that leads to this collection being used. So when you start looking at the releases and the information for these species, I started realizing that the agricultural increase that's associated with this restoration at this scale is really affecting the way that those red dots are placed on the map. In contrast, again, to what I saw in California, where there were more local collections and direct seedings, this system, these Great Basin systems, are so large that to do restoration, you need this increase on farm settings. That's absolutely true. But my suspicion was, when I was looking into the literature, is that it's sort of the tail wagging the dog here, that the requirements for agricultural production were actually trumping the needs of land managers on the ground. And so last year, my student, Owen Boffman, went through, he went through data from 56 different cultivars that are often seeded in Nevada and surrounding areas, and he really quantified the traits that were prioritized during the selection of that particular cultivar. So, this is the reason why that red dot is on that map. And it was actually surprisingly hard to figure that out. Often the plants are just described, but it doesn't say we picked it for this reason. Sometimes it does, but often it doesn't. So he went through a bunch of these releases and just assumed that if these traits are being talked about, they must be important. And so looking at grasses, forbs, and shrubs, the number one reason why, or the number one mentioned trait about these things was this top one, forage yield and quality, being large, above ground, and having good forage yields. The next one, seed yield. Again, this is an agronomic consideration. Seedling vigor was number 3. That is not typically defined, but I can only assume it means being a fast-growing, large, above-ground seedling. It's only when you get to number 4 and 5 where you start getting to things like drought tolerance, or down here being competitive with annuals. Things that managers might actually want tend to be mentioned less than these things that are perhaps appropriate for pasture grasses, right? Forage yield and quality might be exactly what you want in an irrigated pasture, but I do not need to tell you that the Great Basin is not irrigated, and this might not at all be what leads to success on rangelands. Same with seed yield. Seeds are great and they're necessary for farming and for this increase, but they might not at all be what plants need to survive in the desert. So the work that I've been doing is trying to answer this question. If I don't particularly think that these traits are the best traits for cultivar selection or restoration material selection in general, what do I think we should put in that list instead? And that's where the field experiments come in. rather than just putting my opinion, about what I think, even though it's funny, the list I came up with, actually my opinion probably would have been pretty good. But instead we, put a lot of boots on the ground and undergraduates to work and did a lot of testing to answer this question. So I'm going to talk about two sort of major threads of this research, and it's all sort of focused towards learning from natural selection. These plants face a lot of pressures out there. It's really hard to live, and you can learn from the traits that help plants establish in these super hard situations. I can examine directly seeds that survive in these restorations, and I can ask, what traits do these guys have relative to all the seeds that didn't come up? And I can learn from that what helps you grow. The other method that we've used is looking at traits of plants that are surviving in these invaded and disturbed systems. They're naturally out there, and I can sort of interview them, if you'd like, and see what kind of traits the current plants that are succeeding have relative to ones that have gone away. So first I'm going to talk about the first question, looking directly at seeds that survive in restoration plantings. And really asking this question, when we have a giant seeding and we get somewhere between 1 and 5% of seeds established, is that establishment random, or is that establishment based on survival due to specific traits? Right? So do you live because you happen to land in the right place at the right time and you have the right amount of water, or are you living because of something that you are doing? And that is important, and I'll tell you why that's important. And the way that you answer that question is that you basically compare the seeds that are going into the ground with the seeds that are coming out, and you ask if they're different. Let me elaborate on that a little bit more. This is work I did with my master's student, Sarah Kulpa, in 2 fires that burned in Elko County in 2006. And these were seeded by Nancy Shaw and her collaborators, looking at different seed mixes and drilling types. And at this point where she was putting these tests into the ground, I called her up and asked her if she would just save a sample of the original seeds that she was putting into the ground. So the exact same seed lots, the exact same seed mix went into the ground at 2 different sites. And she saved this original material for us. We were able to go out 2 years later and collect seeds from the 1%, if you will. It was somewhere between 1 and 5% establishment at this site. And we were able to collect seeds from these squirrel tails out here and compare them with this original collection in the bag. I often lose people at this point. Like, it's kind of, to me it's not that confusing, but if you don't get this, you're not going to get my next 6 slides. So at the risk of being really repetitive, I'm going to say it again. The question is, in this bag of seed, is it just random plants surviving out here, or is it particular individuals within here that are surviving? And that's key. The way we did this was we collected seeds from 100 different plants at these 2 fire sites in Elko County, and we compared them to that original grab bag of seeds. I have many species from this site, but we picked one of my favorites, Elamis elamoides, bottlebrush squirreltail. It was the only native that was really making seeds reliably 2 years out after planting. So we brought all those seeds back, the 1%, that actually managed to make it, and the original seeds from the bag, so to speak, and we planted them in a common garden at behind UNR. This is a really dry site, 7-inch precip zone, not a really lovely place to live in any way. And we grew them for 2 generations out here. Sarah measured a bunch of things, seed weights of the seeds going in, seed weights coming off for 3 generations actually. We looked at plant height, leaf number, biomass, looked at total seed production, emergence timing, when they're flowering, and then how much allocation they're giving to roots. Today I'm only going to show you data about the seed size and biomass just for time. And remember this question here, we're really asking, do we see shifts in traits between the original bag and the surviving 1%? First I'm going to show you the seed size data. And I'm going to show you a bunch of histograms that look like this. So on the bottom we've got seed weight in milligrams, and then these are just counts going up the side. The propagation field, this is the original seed, so the seed from the bag. And then these are the seeds we collected directly from these 2 fires, the Gopher and East Humboldt fire. And so these histograms are just showing you the distribution of sizes of seeds. And you can see that the average size coming out of the bag is about 3.8 milligrams, whereas the average size of seed coming off the field is much smaller, 1.8 or 2 milligrams. So we see this shift, this seed size decrease in these field collected sites. That's not too surprising. These were grown on a, these were grown on a farm, and they were given fertilizer, probably, and water, who knows. Anyway, their life, they did not have cheatgrass all around them, I'll tell you that. Whereas these were in a fire and it was dry. So this could just be an entirely maternal environment-driven effect, which is why you need to grow them for a whole other generation, if not two. And so this shows you that F2 generation. So again, we see this nice tightening of the mean. The original seeds are still quite big, 3.7 milligrams, and we see that these seeds that actually the 1% that managed to survive are still a subset. They're still this small seeded subset of the original seeds. So basically what's going into the ground is all of this, and what is establishing is this little part right here, right? So the difference between these red bars really indicates the degree to which there's a mismatch between genotype and environment. Ideally in restoration, this would be exactly the same. That means that what's going into the ground is appropriate for your site, but when you have this kind of gap, this is money. And when I talk about this with people who care about that, it's like the distance between these red bars is money that is being wasted, basically. And if we could pull them together, that would be really the thing to do. Like I mentioned, we did an F3 as well, and we see the same thing. Larger means and then smaller and almost identical means of this 1% that's surviving. So this is really cool, I think, and this is consistent with natural selection for smaller seed size in this restoration setting. It's not just seed size, it's also total size. And so here, again, this is the F1 generation. This is total plant size, in it says count, but that's actually biomass, sorry. And it's showing you the same pattern, that the ones that are surviving are small and the ones that went in are big. And that's the same in the F2 generation. Okay, so to summarize that part, survival in our restoration was not random. We saw plants with a correlated suite of traits had increased survival. I showed you that they were smaller plants and they had smaller seeds. They also made their seeds earlier, they emerged earlier, and they had greater root allocation. This is really useful information because, as mentioned, if you wanted to optimize dollars per seed at this site, you would be best served by targeting a seed source that has these kinds of traits in them. The other reason why I love this is basically this method is really easy. Every restoration on Earth is basically an experiment. All you have to do is save some of the seeds that are going in the ground and then compare them to who survived, and restoration can be optimized across precipitation gradients or invasion gradients. This really could be used to make every restoration as efficient as possible. So I like that method. Just a note, in case you haven't thought about this too much, which I had not, when I first started seeing these results, it's possible for small plants to have higher fitness than large plants. Everybody always thinks that bigger is better, but that's actually not true, especially in drought systems. There's evidence that smaller plants can actually be more drought tolerant than larger plants. That's true in both field studies and also if you think about agricultural systems, there has been deliberate breeding of dwarf varieties of certain crops because they're more drought tolerant than large varieties. There are cases where being small is good. Another little note, we did an experiment looking at native plant size over time using a bunch of annuals from the Great Basin in the herbarium, and we found that as the climate has been warming, native plants actually have been shrinking over time. Other evidence from some of the seed zone work that's coming out of the Forest Service, they're also seeing correlations between plant size and aridity, with smaller and smaller leaved plants being more common in dry situations. So if you think back to that graph where I showed you, the traits that we're picking currently to put the red dots on the map for restoration selection, being large and having large biomass yield may be exactly the opposite of what we need in native plants to be drought tolerant. Okay, a second part of our research has been looking at traits of plants that survive in invaded and disturbed systems. These are really fun natural experiments, actually. Sorry, just took a drink of water. And this was, again, sort of inspired by moving here from California. I know nobody likes Californians and Nevada, but I have to tell you that sometimes it's nice to have a fresh pair of eyes looking at these sites. And so this is a picture of Peavine Mountain, which is right outside of Reno. And, just driving around, it's a highly disturbed site. Burns all the time. It's full of shotgun shells. It's not a preserve in any way. But what I noticed is that I came out here expecting everything to be cheatgrass all the time. And in fact, there are tons of native perennial grasses, even in our most disturbed sites. And hold on, there's skateboarders outside the window. I'm going to close the window real quick. Sorry about that. It's lunchtime. So anyway, when I was wandering around these systems, I was joking that if this was California, we would put a fence up around this and call it a preserve, right? Like this is the most beat up part of Reno, Peavine Mountain, but it has more native plants in it than some of the remnant prairies I've seen in California. And so looking at this and looking at these natives making it in this sea of cheatgrass, I started wondering if this is just sort of the last gasp of this system. Are these plants going to wink out? Or are these sort of a specially adapted subset of what used to be in this site? Are these plants here because they're really good at growing with cheatgrass? This is really an evolutionary question. I'm asking, just like in my previous experiment that I talked to you about, I'm asking if gene frequencies have changed over time in these populations. If you think about the bag, I'm asking about if the ancestral population had different suite of traits than this sort of remnant native population. Unlike that last experiment, I don't have Nancy Shaw around to tell me what these plants were doing 200 years ago. I don't have a sample of what used to be here. And so asking if these are these unique requires a comparison to something. And I had to think about that a long time to figure out how you would know. And what I came upon was this sort of method where you could study invasions along these boundaries. So this is a fire scar. This burned in 2006, and it was stopped with aerial fire retardant drops and fire breaks. And you can see on this side, there's significantly more cheatgrass coming in than there is on this native side. And what I realized you could do is you could treat these kinds of old fire boundaries as sort of natural experiments. If you treat this whole thing as one ancestral population, there's lots of gene flow in here, I can ask if this group over here that's experiencing a lot of cheatgrass competition is different from plants over here, right? So again, this would be my 1%, and this would be my initial population. So we realized using this method, you could sort of infer what used to be there and ask if there's been evolutionary change. We found 5 of these kinds of locations in the Reno area, and we tried to find areas that were along roadsides where those fire scars were likely to be persistent and old. We used herbarium records to find places where cheatgrass had been present for at least 40 or 50 years. And so it's kind of hard to find these spots, but we did find several of them. We focused this on 5 perennial grasses. These were picked because they were the plants that were able to make it in these invaded systems. So squirreltail, Poa secunda, Thurber's needlegrass, Hesperos stipa, and Achnatharum. We did this work using transplants from the field. So we would go out, in this case this is an invaded area, and we would dig up usually dormant bunch grasses, and we would bring them back to the lab. We would always treat them really nice. This makes me laugh. Like, this plant does not care that we're squirting it with water at all. This is like a super tough Poa secunda, but we are just giving a gesture here that, like, we care, right? But what you can see here is that this is one individual, and we can split him up into a bunch of different clones if you want. It's a very handy way to get clonal plants. And so this work, we're asking 2 main questions. First of all, that evolutionary question. Are plants from these invaded locations more competitive than their plants from sort of the naive population? Is there something special about the 1% that's making it? And the second question is why? not just are they different, but what traits are associated with any kind of increased performance. And so we did this work in the greenhouse. These are our little senescent, excuse me, plants that we transplanted into pots. We brought them back to the greenhouse, split each plant in two, and we planted half with cheatgrass and half without. So you can see here, this is plant 156. This is growing on its own, and then this is the same genetic individual over here, but he's got cheatgrass crowded all around him. We let them grow for a growing season until the cheatgrass was starting to senesce, and then we harvested them. And here you can see this guy now with all the cheatgrass removed. He actually didn't do that bad. He was able to still keep green and grow despite this competition around him. And what we did for these individual plants was compare how well they did, right? Like how much of a decline did they suffer from being alone versus having cheatgrass with them. And so this is the types of data that we would have. So in this example, this represents one plant. And so this is how many leaves that plant had when he was grown without competition. So like 25 leaves. And then when I planted all that cheatgrass around him, he went down to like 10 leaves. So experienced about a 65% decline. Compare that with this plant over here, planted him alone. It's 20 leaves, plant with competition. He doesn't even really notice, just a 17% decline. The reason why I go through all the work of showing you this is because in biology, usually bigger numbers are better. But in this case, 65% decline, the higher the number, the worse you're doing. So keep that in mind in the graph I'm going to show you. And so the point of all of that work, those transplants and those competition experiments, was to answer this question: are those plants from the invaded areas more competitive than the plants on the other side of the road? And what we found was that in 3 of our 5 sites, that was the case. So here in gray are plants collected from invaded areas, and in blue are plants collected from these sort of ancestral or uninvaded populations. And remember, this is a circumstance where a high number is bad. So the blue, sort of the ancestral population suffered a greater decline in size than the more experienced plants growing on the invaded side of the road. This was true in 3 out of our 5 spots. In one spot, the opposite was true, and I sort of have a theory about why that might be, but I won't go into it here. But I think the point from this is that in some cases, you really would do well by collecting seeds from this invaded population on the other side of the road. In other cases, not so much, but I think it begs the question of, calculating this and thinking about this kind of performance when you're doing your seed selection for restoration. You don't have to select seeds in ignorance. You can know that the population that you're picking has a really good ability to tolerate cheatgrass competition, and I think that's pretty key. I'm going to show you a breakout of variation within this one Balls Canyon site, which I also think is pretty cool. This is cheatgrass weight. What I was showing you before was how the native plant was doing. This is an index of how much the native plant is affecting cheatgrass. And in this situation, each bar is an individual maternal plant from the Balls Canyon site. And you can see that there's huge variation in how happy cheatgrass is. So down here, these native squirrel tails are really suppressing cheatgrass a lot, whereas up here, the cheatgrass is actually being facilitated. When it was grown on its own, I think it was about 2.5 grams, and it's actually doing a lot better when grown in competition with some of these. I've color-coded the bars blue and green, blue and red, and the interesting thing here is that the red families are from the invaded sites, and the blue families are sort of the ancestral population. And I like looking at this because what I see, it's exactly what you would expect, the pattern from natural selection. In the blue ancestral, there's variation, and there are some of these highly competitive types, and you can see the signature of selection here. In the invaded area, you're getting mostly these competitive suppressive genotypes and only a few that can't suppress cheatgrass. So I love it that there's this variation in the wild and that you can see selection happening on the landscape. From a management perspective, again, you're going to want to be choosing the populations that have these kinds of families in them that can really suppress cheatgrass over these who are, if anything, facilitating it. Okay, so that's the first question. Sometimes, yes, you can find these really competitive plants growing in invaded locations. The second part of this question is why, right? Like, what are the traits that are increasing performance? And this is the slide that I often show, and I don't go too much further in terms of showing why. But this time I'm going to tell you what we've learned, and I'm going to show you a little bit more of the why we think this is true. So these are characteristics through a whole bunch of field and greenhouse experiments that we think should increase native performance when grown in cheatgrass invaded sites. So a bunch of things about phenology. Greening up early in the fall seems to be really good. Flowering early and germinating early from a seed. These all seem to help you when you're competing with cheatgrass. Growing lots of roots early on. The sort of decisions you make about root morphology matter. And then also, as mentioned, being small, both adults and seedlings. I'm going to show you a couple examples of why we think early green up is good, and I'm going to show you a couple examples about early root growth in seedlings, which also seems to be really important. Okay, so phenology. When we dug up these dead plants, I keep calling them dead, they were senescent. When we dug up these senescent plants and put them in pots, it was really obvious that some of them responded much quicker to water than others. And I did not have any expectation of what was going to happen when we added water to these plants. I didn't know if they were going to take months to green up or if they'd green up instantly. What was really obvious is that some particular plants that we collected out of the ground grew leaves like faster than you would possibly could have imagined. And so the first time we did this, it was with a squirrel tail collections from these invaded and uninvaded areas, and we just added some water. And I didn't know that this was going to be important, so I didn't track when they were greening up. But just at 10 days after adding water, it became very obvious to me that some plants were greening up and some weren't. So over here you can show this is just the percent plants that were green 10 days after I put them in a pot and added water. And 80% of the plants from the invaded sites were green. And only about 40% from the uninvaded sites were green at 10 days. They would all eventually, this would get to 100% for everybody, but there's been this real shift towards super early green up. My next collections, we were sort of cued in that this might matter, and so we actually counted the days to green up for these different ricegrass and squirrel-tail collections. And again, here we've got invaded populations in gray and the uninvaded sort of ancestral populations in blue. You can see that the plants from the invaded are greening up, a week to 10 days before their, plants from very close. This is literally across the road. So in every phenological example, we've seen early green up in these invaded populations. It actually matters in terms of their ability to tolerate cheatgrass competition. So if you look at this one experiment with squirrel tail, for example, if I look at this percent decline in size, this is that circumstance where a high number is bad. If I look at if they initiated growth early or if they initiated growth late, the plants that initiated growth early had less of a decline in size, only about 50% relative to about 75% decline in size. So you can see there's evidence that there's been selection for early green up, and that even in these pots, that kind of phenology shift is really translating into ability to grow a cheatgrass. And this makes total sense to me. Cheatgrass is an annual. It has to grow this whole root system. But if you're a perennial, the minute water falls from the sky, you green up and start sucking it from the soil, that is a really good strategy, right? I mean, it just makes sense. So phenology seems to be really important for competing with cheatgrass. The other thing that we spend a lot of time looking at are roots. And that's because in these dry desert systems, the contested resources are below ground. All these plants have plenty of light. You do not need huge, leaf systems to shadow each other. That's not, this isn't the tropical rainforest here. They all have plenty of light. The resources that are limiting in deserts are below the ground. There's often a misperception about cheatgrass. People will call it a shallow-rooted annual. That is not true at all. This is an excavation below a cheatgrass monoculture, and this is a meter stick. And these are cheatgrass roots going down to a meter and well beyond. Don't be fooled. If you pull a cheatgrass from the surface, it breaks off really easy. It has very fine roots, but those roots go down really far. And anyone who's ever grown cheatgrass in a greenhouse can tell you, man, it produces huge amounts of roots, and they go deep very quickly. And so for any native perennial who's going to be planted into the system, they're going to have to be able to deal with the fact that cheatgrass roots are everywhere, and they're deep, and they grow quickly. Root work is most readily done in greenhouses. Actually, so we've done a lot of this sort of screening of root traits in these big sort of pot experiments in greenhouses. We have done experiments where we've grown plants for different ages. We've harvested natives at 10 days old, 50 days old, and 100 days old, and sort of wash soil from roots and sort of measure them. And we can sort of describe the root traits and then ask how well these root traits predict success when we plant them with cheatgrass. The really good thing is we found that what plants do when they're 10 days old matters a lot. The reason why that's good is because it's really easy. This is like probably a 50-day-old squirrel tail growing with cheatgrass, and this takes like 45 minutes for some poor undergrad to get that situation cleaned out. But what we've realized is that these 10-day-old seedlings tell us a lot about success, and these are a lot easier to work with. This whole thing is a scan. It's probably only 4 or 5 inches from tip to tail. And you can get a lot of information here, from leaf information, seed information, root to shoot allocation. We're interested in the length of these roots, how fine they are, the average diameter. We count, well, we don't do it. We have software that does this. Counts the number of tips, which are places where water and nutrients are absorbed. Anyway, you can get a lot of information out of a 10-day-old seedling without too much trouble. And the reason why this is important is in our experiments we found that early root growth, what these plants are doing in those first 10 days of their life, is the most important predictor for how they're going to do with cheatgrass. So these are results from a pot experiment, container experiment, where we looked at those root traits and how well they correlated with the ability to, whoops, oh my gosh, what happened there? Their ability to tolerate cheatgrass competition. And these, this keeps changing on me. I'm so sorry. What we found was that the correlations were strongest between these 10-day root traits. Those 50 and 100-day root traits were not as powerful at explaining success. I'm going to show you this one, the example of how many fine roots you've made at 10 days and your ability to tolerate cheatgrass competition. So here we have the length of fine roots in centimeters. Again, these aren't huge numbers, We're varying between 1 centimeter and 4 centimeters here. And this is how competitive they were. And I've made it so that a high number means that you were very competitive, just so it's intuitive. And you can see that there's a pretty decent correlation here between making fine roots very early on and your ability to tolerate cheatgrass competition. So that's cool. But that's from a pot, which is very different. And we what we're trying to do the last, I would say, few years, we've moved from those pot studies explaining root traits and trying to figure out what's going on to asking how these root traits affect performance when we plant seeds out in the field. And so this is a situation, yes, it's still unrealistic, it's still nerdy science. We're still tracking individual seeds, which we do by gluing them on toothpicks. We really want to be able to seed into the existing community, be able to follow our grass and ask how it does. And sort of another slight subtlety between what I've been doing the last few years and the information that we gathered previously is that I'm sort of moving from looking at traits of individuals that help plants survive to traits of populations that I'm sort of trying to identify populations with a high proportion of these successful individuals. And the reason why is, I've learned a lot from plant 74. This plant told me that green up, early green up is really important, but I don't want to just, I don't think that the Great Basin should be full of the progeny of one successful individual, right? Like, I don't think that we should find the most important individuals and increase them for restoration. Restoration really most naturally happens at the population level. We need to preserve diversity in these systems because, first of all, cheatgrass is not the only weed that these plants are ever going to see in their lives. We need to maintain diversity and the ability to respond to other things that come. Climate change is happening. We we do not need to hamstring our native populations by, giving them just this tiny little bit of diversity to work with. So I'm not comfortable with that, but I am comfortable with testing populations and trying to find areas where we can do large-scale, hopefully diverse collections, but places where we have a high proportion of these successful plants. So that seems sort of like a subtlety, but I actually think it's really important. And so the sad part about that, it means you have to drive more. So all the other work we've been doing, we could do local, and it was really sort of tests of concepts. This really, you need to get in your car and do some collecting. So my postdoc, Dan Atwater, and some undergrads here from UNR went driving around. This is 2013, I think, is when they did this, and they collected seeds from 35 squirrel tail populations around the Great Basin. This was a really dry year and a really hard year to collect seeds. So they visited more populations than they were ever able to get seeds from. Their target was anywhere they could get a good collection from 70 to 100 plants per population. So we brought all those seeds back to the lab, and we first described the variation in the seed and seedling traits that I found to be so important, right? So I had undergraduates measure seed size. We looked at time to emergence in a greenhouse. We grew them to 10 days old and then scanned them for these root traits that we found to be important. We also looked at total plant size and this root to shoot ratio. And just to note, this was such a dry year, we sampled only about 20 seeds per population to describe the seedling traits, which is pretty ridiculous and like way less than you should do. So if you have the ability, you should do more than 20. But honestly, we were so seed limited, we couldn't spare more than this. So this small sample, when we're trying to describe, the entire seedling trait of a population, this sort of only makes the results I'm going to show you even that much more spectacular, because it turns out we can even learn something from 20 seeds. Not that I'm suggesting you should. So the first thing, the good news is that there is variation. These are our 35 populations. And I've just graphed two traits here, how long the roots are at 10 days old and how thick they are. And you can see there's a lot, these populations have long, thin roots. These populations have long, fatter roots. There's a lot of variation out there in squirrel tail in what it's doing with its roots at 10 days old. And what we're really interested in is, does this kind of variation predict success in the field? Is this the kind of population you want, or is this the kind of population you want? Where along this spectrum are the plants that are going to do the best? So we took these wild collections and we planted them out at these 5 dots throughout the Great Basin. We put 50 seeds per population per site, so almost 2,000 seeds per site. Planted them in the fall. These are highly disturbed systems. This is Paradise Valley. The mountains and the sky are beautiful, but the plant community is fairly sad, I would say. It's cheatgrass. There's some Poa secunda in here, some weedy forbs, but not much in the way of native diversity. We sprayed these sites the year before to knock back the cheatgrass seed production, which you pretty much have to do when you're seeding into this kind of environment. And then we seeded on toothpicks directly into these invaded sites. There's still plenty of cheatgrass here. Don't worry, this wasn't comfy for these plants in any way. I'm going to show you just 2 different performance metrics. Whoops. Oh my gosh. Oh, that's rude. Sorry, my PowerPoint just died. Just did the good part too. Oh, hold on, I'll get there. That was funny. Sorry for the inconvenience. Yeah, that was fairly inconvenient. Okay, so we put them in the ground. Oh wait, you're looking at the wrong thing. How's that? Good?
Yeah, that's good.
Thanks. So I'm looking at just 2 different metrics, whether or not the plant was present in June and if it was actively growing in June. Sometimes they were happy. I would say rarely they were happy and green. Often they were just totally absent, and sometimes they were present and very, very brown. So we really want to know, do those 10-day-old root traits predict your presence and your sort of photosynthetic happiness in June? And so the good news is that, yes, there was a lot of variation among populations in whether or not you were present in June, from about 17% at the low to about 45% at the high. You see even stronger population differences in sort of your photosynthetic activity in June. I kind of think of this as like how happy you are, and probably where your roots are. If you're brown, you probably didn't get your roots that low. But if you're green in June, you probably have access to water. And so what you can see is that some of these populations are doing really poorly, but there's this chunk up here that's doing pretty darn well. And so from a restoration perspective, these populations down here, they might have all kinds of agronomic traits that make them really suitable for seed production in some kind of field. But what we really want to know as a manager is where up here are your most successful plants that are also really good at, performing in an agronomic situation. I feel like this kind of test should be the first screening for how to put a red dot on the map. And then we should start looking at these and maybe only work with these and say, of these, which one is most amenable to agronomic increase? Do you see what I'm saying? You can't talk to me, but but anyway, you get my point, right? And what we're doing now is we're not at all creating this kind of profile of our seeds. They're not being screened for whether or not they can grow in Paradise Valley, Nevada, in the middle of a cheatgrass invasion. They're being screened for very different things, and I think that this method is really important. Okay, so that's my soapbox. The question is, okay, so there's variation here. You could just leave it at that, and you could just screen these populations, but I'm also interested in knowing why some populations are better than others. And the cool thing is that these seed and seedling traits that we screened for these 10-day-old seedlings did a ridiculously good job predicting success. So here we have your percent presence in June for the whole population across all 5 sites, by the way. This is just a very coarse look at the data. And this is how long your roots were at 10 days old. And that is a beautiful line, you guys. This tells you that 50% of the variation in whether you were there or gone in June can be explained by this one root trait, right? So again, you can screen plants and see who can grow these really long roots at 10 days old. And these are the kind of plants that we want to be selecting for increase. When you look at the whole model, when I put all of my traits in there, I can get that R-square really high. I can explain 64% of the variation and whether that seedling is going to be there, just looking at root length, seed weight, and the number of root tips. The interesting thing about seed weight is it's not a direct effect of seed weight on success. It's an indirect effect. So if plants with large seeds use that energy to grow long roots, they're more likely to survive. So if you put this into a path analysis, it tells you that seed weight is not direct, it's indirect through the growth of long roots. So that's really cool. So that's actually my last data slide. My summary here, I love thinking about every restoration as an experiment. If you just save seeds that are going into the ground, you can learn from every experiment and you can try and optimize your mix and your conditions. Try to get those red lines as close and tight as possible. I also think this point that invaded populations can be really good places to find competitive plants is a point that I think is worth making. I also feel that field testing performance is the most important way to choose restoration sources for seed increase. Yes, it takes time. It adds a whole other year, to the seed increase work. But I'll tell you, it can engage a lot of undergrads in your work, and they do like working on these issues. They do like feeling like they're making a difference. And it could lead to, considerably different, I'm going to go back, I told you I lied, but look, I mean, if you spend that year and your difference between 1% actively growing and, 9 or 10% actively growing, that's a significant, increase in success. And taking that year out to do that kind of study, I think, is worth it. And, you might find that these traits that you would not suspect, being small, making lots of roots, early phenology, you can, apply this to any system. You can directly test what suites of traits are helping plants survive in the field. And with that, I will thank my funders and a lot of collaborators. Here I've listed postdocs and grad students. There have been tons of undergrads who've paid their way through school by rinsing roots and gluing seeds to toothpicks, and I couldn't have done any of this work without all of the support of all of these. And I'll thank you guys for listening, and Janie, I will take any questions that you can pass along.
Okay, great. Thanks, Beth. So if you have questions for Beth, please type them into your questions pane in your control panel. Lindsay Robb asks, and this is regarding kind of your introduction towards the beginning, where you had watered seeds and then turned the water off the plants and they died. And so she asks, are you just simplifying when you say you turned the water off, or did you do a gradual hardening?
I guess I am simplifying. So we watered them through the sort of natural growing season and into spring for the first 2 years, and then they went into the natural winter. You know, they got their normal winter precip. The only difference is we didn't then supplement it come following spring. So they should have been able to harden off at that point. They should have been able to put roots down and persist, but they did not. No.
Okay, great, thanks. Steve Popovich asks, regarding the first set of 6 slides you showed of examining random selection seed attributes over time, the 2F2's seed character means you showed in bar graphs from the 2 fire rehabs were so similar to each other but different from F2 control seeds. Were the 2 fires on similar sites, ecosystems, soils, or in very close proximity to each other? Wondering why they were so similar to each other in seed attributes measured. You'd think they'd sort out different between fire rehab site conditions. Any speculation?
Yeah, so that's really interesting. Those 2 fire sites, I think they were about 60 kilometers away from each other, so 40 miles away, and they were similar soil types, but they had very different amounts of cheatgrass. So one of them, the Gopher Fire, had tons of cheatgrass, and the East Humboldt Fire had much less. Yet, even given that, it looked very much like this very targeted subset of that seed survived. And I agree with you. Those means are remarkably similar. I never did any neutral molecular DNA testing on these, but I have thought about it. I mean, what if that was really just one particular family that can hack it out there? So I really don't know how much variation there is in that sample. But yeah, I noticed that too. It's a remarkably similar group of plants that survived at both sites. And I don't know what to tell you about that, except for I think your instinct is good. What's going on there? Yeah.
Great. Thank you. So you have a lot of thanks, great presentations, and then someone asked, hopefully, or has stated, hopefully you've commented on the proposed national seed strategy.
Yes. Yes.
Great. Excellent. Steve follows up by saying, this is important stuff and stressing local native plant materials is ramping up with Fed agencies. Are you getting this info out to our seed community? Native seed meetings like, at Santa Fe, which you were at, sharing with major producers and seed companies, regional botanists, etc.
Yes, I really try to give this talk or some version of this talk as many times as possible, and I do feel like I have gotten the information into the right hands. I feel like this is a battleship that takes a long time to turn, but I now go to meetings and I am not the only one saying that small plants are good, and I'm not the only one saying that we should think about roots, and that makes me really happy. Whether or not the seeds and the seed warehouses have changed, I don't think that has happened yet, quite honestly, but what I'm seeing instead are local offices, local BLM offices deciding to sort of go their own way and collect seeds from their own local communities from plants that are important for them, and contracting their own seed growouts and maybe sort of bypassing this sort of cultivar selection, which I think is actually really awesome. And I think one of the collaborations I'm doing with the Winnemucca BLM, they're doing a Seeds of Success collection this summer, and I told them, I have some money hiding in my couch cushions. I'm going to find some undergrads to screen their populations for 10-day root traits. That'll only take me about a month. And then I can give them that information and say, okay, of your 10 collections, these 3 are the ones that I think are worth, increasing. And so I think we're doing it a little bit grassroots, but I think things are changing. Yeah.
And just my own question following up to that, do you think, is there a place for major producers and seed companies? Could they get involved in this?
Absolutely. I think, like I said, I think the thing is to screen populations for rangeland function first, and then I am very happy to hand someone, 10 collections and be like, see which of these is most suitable for agronomic production. I think it's just the swapping, we should not wait and find out how they do on the rangelands at the point where they're really being produced at these large scales. Or, rangelands, they might do fine in Oregon and Idaho. I'm really talking about Nevada and the places where I live and work, where some of these larger plants just do not grow. But we cannot do it without large producers because the scale of these restorations is so big you can't simply do wildland collections. It's not enough. I think my point is really think about why you're putting those red dots there before they get agronomically increased.
Great, thank you. Well, that looks like the last question. There are a lot of thank yous, which I'll send those to you after the webinar.
That's very nice.
Yeah,
well, it's very hard to not see you all, and thanks for listening. I can say that.
Great. Yeah, so we would greatly appreciate it if you would take our 3-question survey of this webinar that will appear after the webinar has ended. And a link to the recording of this webinar will be posted on the Great Basin Fire, I'll post the video on the Great Basin Fire Science YouTube channel just right after the webinar, and then you'll get an email tomorrow from the GoToWebinar system showing you where the link, where you can find that. And our next webinar in this series, titled Weather Variability and Forecasting Tools for Short and Long-Term Restoration Planning, presented by Stuart Hardigree, will take place on May 12th. If you have any more questions regarding this webinar or have requests for future webinars, please call or email me at any time. Thank you all for your presentation today, and thank you so much, Beth, for your presentation.
Thank you.
I think I might have thanked everyone for their presentation, but I meant participation. Sorry, I have a little bit of a cold. Okay, great. Thanks again, and have a great day, everybody.